Okay, so hi everyone. My name is Jeanette Cabrera, and I will be discussing my journal club article with you all. And the title is Soil Microbiome Responses to Extreme Climatic Events. And here I provide the full citation, including the link. So let's get started. So um, the research question and hypothesis. So the researchers aim to find out if there is a unified microbial community response to extreme climatic events across soil types and biogeographical regions. And there were two hypotheses put forth, and the first was that the authors expected to see similar disturbances to shift soil microbiome communities in the same direction. The second hypothesis that was put forth was that local climatic conditions select for soil microbial taxa, exhibiting traits that allow them to cope with extreme climatic events regularly occurring in their climatic niche. And the goal achieved? Yes, the researchers were able to determine the severity of microbial community responses to extreme climatic events that relied on the original environment. The important contribution was constructing a predictive model for the impact of extreme climatic events on soil functioning. So again, the goal was achieved and they were able to actually come up with a predictive model. So why should we care about this field? It is important to provide critical understanding how ecosystems behave under climatic stress. This can help with anticipating shifts in biodiversity or planning for ecosystem collapse or transformation. We can then determine how to support recovery and adaptation through land use and restoration. So I first want to start off the state of the field for my background. So soil microbial ecology has increasingly focused on the resilience and adaptability of microbial communities to environmental changes, especially in the context of climate change. Previous research has highlighted the sensitivity of soil microbiomes to factors like temperature fluctuations, moisture variability, and extreme weather events. However, the predictability and consistency of microbial responses across different ecosystems remains less understood. So I actually was able to find some articles about what is the state of the field, and this is where I got this article from Bardet, um, that again focuses on exploring mechanisms of soil microbial communities. And the broader research context that I want to add for you guys for this background is this study's approach of analyzing soils from 30 grasslands across Europe under controlled conditions provides a robust framework for assessing microbial resilience and adaptability. So that is something new. Usually, it's only the robust framework of different soil analysis for assessing microbial resilience is new because before there was only single soil or just a system, not different like the 30 different grasslands. And I was able to find an article that led me down that path. And they underscored the importance of incorporating microbial dynamics into ecological models and climate projections. So, what does this need in soil microbiology? So, the broader implication is the implications of this research extend beyond soil microbiology. Understanding microbial responses to climate extremes is crucial for predicting ecosystem functioning, nutrient cycling, and carbon sequestration under changing climate conditions. And why is this urgent? Human health, food security, and economies depend on stable ecosystems. Having a deeper understanding helps to prepare for impacts of extreme climatic events on agriculture, fisheries, and water supplies, and can guide policies for sustainable resource management. So, going into the article, what did the authors set to answer? They wanted to see, the researchers set out to answer if there's an integrated microbial community response to extreme events such as drought, flood, freezing, and heat across soil types and regions in Europe. So I just want to now move on into the methodology, and I'll first provide the critical details that of the methodology that were conducted. So to start off, 30 soils from different European grasslands underwent exposure to heat, freezing, flood, and drought for 2 weeks alongside a control. The soil microbial community response at the end of each said condition was measured by fungal, prokaryotic, and shotgun metagenomic sequencing. And soil functioning was evaluated by measuring enzymatic activities, substrate ability, and the amount and movement of carbon and nitrogen in the soil. So, definitely going through this article, I found myself searching up the definitions of different words or concepts. I wanted to share with you all these words and concepts that were specifically new to me and might help you. You can comment and see if you already have heard of these terms, if these terms are also new to you, and that would be a good way to show, like how this article is able to provide new information or is presented in a new way that you haven't seen it before. So the first word is biogeographical region, and this was defined as areas with distinct biodiversity of animals and plant species. The next one that I had not even heard of at all was microcosm. Don't know if I'm saying that right. It's a small simplified control system that models a larger system, so that does make sense. And amplicon sequence variants are also known as ASVs, and this is how I abbreviated it in the next following slides or throughout this presentation. And these are amplified sequences that are error corrected that generally represent a distinct microbial species or strain. So, yeah, those are the three words: biogeographical region, microcosm, and amplicon sequence variants. So, anchoring the techniques to like what we have learned in the class, we have specific modules in our class that have covered 16S rRNA and Illumina. For my specific journal club, the 16S rRNA genes from prokaryotic and fungal communities from the soil samples were amplified by the V4 and V5 region. And sequencing was performed using a two-step Nextera approach on the Illumina MiSeq platform with V3 chemistry to determine the functional gene composition of the initial samples and microcosms at specific time points. The whole genome was sequenced. The Illumina unamplified fragment library was prepared using the TrueSeq PCR free kit, and Illumina adapter sequences were detected and removed. And here I have included an image of the Illumina MiSeq system here. It is on usually the benchtop, and it looks like a simplified interface system here. And yeah. And now moving on to the statistical approach. Now in examining the statistical approaches implemented in this article, the R software was used for core analysis and plotting. I did want to highlight that the Permanova was performed to quantify extreme climatic events effects and soil functioning measures. specifically testing the effects on sampling time, disturbance, and interactions with the soil site. And now we'll be shifting to the results portion of this discussion. So figure 1 is showing the consistent microbial community shifts across diverse European soils in response to the simulated extreme climate events. So part A is the sampled site locations, and the 10 circles represent the countries where 3 replicate grasslands sites within 11 kilometers of each other were sampled, resulting in 30 sites total. And the sites represent the diversity of biogeographical regions present in Europe, which is the Alpine, Subarctic, Arctic, Atlantic, Boreal, Continental, Mediterranean, and the steppe climate. And part B is the experimental setup for the simulation of extreme climate events. So from these 10 countries, with 3 replicate grassland sites, they took 100 grams of soil, and then this was just the initial measurements, and then they had a 1 week acclimatization period, and then followed by the disturbance that was put on the soil for 2 weeks. And we did have a control, and here are the different disturbances: drought, flood, freeze, and heat. And then followed by a recovery period, which was a month. And here are the different time points: S1, S2, S3, S4. S1, the end of the disturbance. S2 is just 1 day. S3 was 1 week, and then S4 was a month following the disturbance. So here's like the simplified version of, how many samples we have in total. So 10 countries, 3 sites, 5 treatments, 4 sampling times. And again, those are the replicates, so 630 samples. And now moving into the C through H graphs. So C through H shows the simulated climate extremes consistently shift soil microbial communities in the same direction despite their contrasting composition. So specifically looking at C, D, and E. Non-metric multidimensional scaling, or NMDS. We have NMDS1. Ordinations of prokaryotic, which is C, D is fungal, and E is the shotgun metagenome. And this is based on Bray-Curtis dissimilarities, show that the origin of the sample is the main driver of microbial community composition. And the different colors, as seen by this key, is the different countries. And now moving on to F, G, and H. This was taking the partial redundancy analysis, so RDA. The RDA ordinations show disturbance effects on communities after controlling for side effects. So F is again prokaryotic, G is fungal, and E is the shotgun metagenome. And this showed that the communities exhibit consistent shifts in response to individual disturbances. So there's the control. Here's the key: the control: drought, flood, freeze, and heat. And as you can see, there's a shift. These are all for the heat. And there's a shift. Again, heat, shift, and heat, shift. Now moving on to figure 2. So this is where that definition of ASV was important for me to define earlier. And figure 2 is where the taxon-specific resistance and resilience responses with soil microbial ASVs to extreme climate events was modeled. ASVs that are fully resistant form the largest group, which which is the uncolored in this figure, ASVs that showed significant changes either positively, which is orange and red, or negatively blue and purple. There were 10 different ecological response strategies identified, and these are the strategies here below. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So here they are here. And the most common strategy was an increase or decrease in relative abundance that then remained stable. Part A are the ASVs associated with prokaryotes, and part B is fungi. The central tree, shown here, indicates the taxonomy of the 500 most abundant ASVs, with the tips colored by the phylum. So here are the different phylum shown here for prokaryotes, and here are the phylum for the fungi. And the more abundant ASVs are the more intense colors. And the rings surrounded the tree, one per disturbance treatment, as you can see here. Like I said before, purple and blue colors indicate that the ASVs perform significantly worse following disturbance. And like the key shows, the shade of color indicates a statistically significant divergence from the control at the end of the disturbance, followed by a statistically significant change in the same direction over the following month. The palest shades indicate no significant changes at the end of the disturbance, but a significant divergence over the following month. The results showed that in both prokaryotic and fungal communities, showed resistance to flood and heat, were more phylogenetically conserved than to drought or freeze. So again, resistance to heat and resistance to flood were more phylogenetically conserved. And now moving on to figure 3. Figure 3 discussed extreme climatic events that drive significant changes in protein function, abundance in soil metagenomes. The graph A shows the effects of disturbance on each of the 28 highest level functions, and red indicates an increase in abundance following the disturbance mentioned at the top of the column. So here is drought, flood, freeze, and heat. Again, the disturbances that were examined. And red is an increase, and blue is a decrease. The changes in proportion with disturbance relative to the control is shown on the horizontal axis. And what can be taken from this is that genes involved in dormancy and sporulation found here, increase in relative abundance across flood, freezing, and heat, suggesting a community level selection for these traits across a variety of stresses. And heat treatment elicited the greatest number of significant functional changes. And part B shows the number of individual proteins with and without significant effects of disturbance in each of the 28 highest level functions. And on the x-axis, we have the number of proteins. And this is just showing the key here. Blue is no treatment effect. Yes is there was a treatment effect, and gray, no model converging. And as you can see, there was a treatment effect on the highest one was or is this one, this 59% amino acids, carbohydrates, cofactors with like vitamins, again, and like I said, dormancy and sporulation had the highest, 60%, 60 and 61, DNA metabolism and dormancy and sporulation. Okay, so now on to figure 4, which was linking baseline soil and climate conditions to microbial functional responses to environmental disturbances. So this is the final figure that I will be discussing, and this has the rank correlations between initial properties and resistance on the left. So this is initial, um, and resilience on the right of community function following disturbance. It was found that resistance and resilience to heat and drought and freeze were lower in communities from cold and wet climates, whereas resistance and resilience to heat drought were higher in communities from warmer and drier climates. So this makes sense, right? So like if you have a soil that is from a climate with a lot of heat, it already has those mechanisms and those traits that would make it more adaptable to heat. So that is what they found and they proved to be true. Um, and then now part B shows the relationship of soil functional measurements such as enzyme activities, microbial substrate use, gas fluxes, and soil carbon and nitrogen concentrations. Um, and the relative abundance of functional proteins for metagenomes. The imposed climatic events strongly affected fluxes of greenhouse gases and carbon nitrogen availability, but less so enzyme activities and microbial ability to use different substrates. So all of this data revealed a direct link between metagenome structure and soil functioning, which is when metagenomes were more similar. Measured soil functions were typically more similar within the end of disturbance sampling and following 4 weeks of recovery in each treatment. And specifically in 4B, in all treatments, both S1 and S4. So S1 is 4, S1 is 4. Um, the metagenome composition was strongly correlated with enzyme activities and with substrate-induced respiration. So just looking at the different legends and axes here. So here are the different treatments, here initial properties, and then the different things that were looked at. Like I mentioned, the gases, C to N ratio. And the correlation coefficient is here. And again, we have for part B, treatment and sampling time, the S1 and S4, measured functions, again, soil, C and N, gas fluxes, micro respiration, and enzymes. This figure I thought was important because this just solidified that, there was that the metagenome was correlated with enzyme activities. So and with substrate induced respiration. So, those specific like soil sample communities have have these traits, have these different activities that help them, adapt and are resilient and resist certain extreme climatic conditions. So that was the last figure, like I said. Now moving on to the discussion. So, in restating my bluff, the authors in this study aim to see if there is a unified microbial community response to extreme climatic events across soil types and biogeographical regions. Now we do know that there are different extreme climatic events that do alter microbial soil communities in specific and predictable ways. The broader implications are that we have a starting point on how the impact of extreme climatic events on soil functioning can be predicted to help with climate change projections. We still do not know the long-term data of these disturbances on the soil microbiome. That would be a good, follow-up study to examine and like look in depth more of like how long these traits are observed for. And this study addresses the gap by demonstrating that soil microbiomes exhibit consistent and phylogenetically conserved responses to various extreme climatic events, including flooding, freezing, heat, and drought. Notably, heat stress had the most pronounced effect, leading to increased dormancy and sporulation gene expression and reduced metabolic versatility. This was observed again in figure um, 4, 3, and 4. And these different findings suggest that despite the diversity of soil types and microbial communities across different regions, there are common response patterns to climatic extremes. And my simplified conclusion that I again want to state is that soil microbes respond in a distinct and helpful way to extreme climatic disturbances across different soil sample types. And I have provided 5 questions that serve as a starting point for discussion, and please feel free to answer them or provide your own. So the first is, why do extreme climatic events jeopardize the functioning of ecosystems? Why is it important to be able to make general predictions about the consequences of extreme climatic events on soil function? Was there any part of the results that was shocking to you, or was it predictable? Please provide an explanation. How would an experimental design beyond 10 European countries look like at a global level? Is there a research question or different perspective that was not addressed that would have also added value? And here are the references that I used to strengthen this PowerPoint presentation. Thank you so much for listening to my journal club discussion. Thank you for taking the time to read the article. And I look forward to seeing the discussion that comes about this. Thank you so much and have a great day.